Hello viewers, I am K. Devavani, Assistant Professor of Microbiology from Government Degree College for Women, Nalgonda. In this video, I am going to explain a topic on antigen-antibody reactions, that is precipitation reaction. Antigen-antibody reactions, they are highly specific and these reactions are also called as a serological reactions. The serological reactions are of different types. They are precipitation, agglutination, complement fixation, neutralization and the labeled antibody techniques. In this video, I am going to explain about the precipitation reaction of a serological reactions. So, what is precipitation? Precipitation is a reaction occurs when a soluble antigen combines with an antibody in the presence of electrolytes at the suitable temperature and pH. At suitable temperature and pH, when the soluble antigen combines with its antibody, it leads to the formation of an insoluble antigen antibody complex, insoluble precipitate. In the presence of electrolytes and the particular suitable temperature and pH, when the soluble antigen combines with its antibody, it leads to a formation of insoluble precipitate. That reaction is called as a precipitation reaction. The formation of this precipitate is dependent upon the formation of a lattice. The mechanism of precipitation reaction was explained by Marac in 1934. That includes three phases, that is pro-zone, zone of equivalence and post-zone. Graph shows that the increasing concentrations of antigen is added to the same amount of the antiserum in different tubes. The precipitation will be observed most rapidly and abundantly in one of the middle tubes, where the antibody and antigen are present in optimal proportions. That zone is called as a zone of equivalence. In the zone of equivalence, the antigen and antibody concentration will be optimal. So, that leads to a formation of a lattice here in the zone of equivalence, but in the preceding zone, that is a pro zone, where the amount of antibody is excess, the precipitation will be weak or even absent. Even in the post zone, that is where the zone of antigen is excess, the precipitate formation is prevented because of the excess amount of antigen. In both conditions of pro and post zones, the precipitation formation is prevented because of the either the antigen or the antibody will be in excess. But in the zone of equivalence, the lattice of alternating units of antigen and antibody, they form a lattice structure that leads to the formation of a precipitate. That is called as a zone of equivalence, where the amounts of antigen and antibody will be in optimal concentrations. If the precipitation is suspended, then it is called as a floccules. If the precipitates they are sedimented, they are called as a precipitation. If the precipitates not sedimenting and they are suspending in the solution as a floccules, then the reaction is called as a flocculation. The applications of precipitation reactions or precipitation test can be carried out as in qualitative or the quantitative test. It is sensitive for the detection of antigens and less sensitive for the detection of antibodies. Precipitation is sensitive for the detection of antigens, but whereas it is less sensitive for the detection of antibodies, but can be used for the qualitative and quantitative determination of antigens. And here the precipitation test has an applications in the forensics for the identification of blood and seminal stains, and this has an application in the testing the food adulterants. The adulterants that are added to the food can also be tested by using this precipitation reaction. And another one is grouping the streptococcus. The streptococcus organism is grouped into different categories serologically by means of a technique that is called as a Lansifield technique. In this Lansifield technique, grouping of streptococci into serotypes, application use of precipitation reactions. So, precipitation reactions are also useful in grouping the streptococcus into the serotypes. That was used by Lansifield. So, this is called as a Lansifield technique. Precipitation test has an application in the VDRL test. VDRL means Venereal Disease Research Laboratory. This is the test used for the identification of an organism, that is a Treponema pallidum, which causes syphilis, which is a sexually transmitted disease. To identify the organism that caused the syphilis, VDRL test is generally used to standardize the toxins or the toxoids. These precipitation reactions are useful. These are some of the applications of precipitation reactions. And next, moving on to the types of precipitation reactions. To form a precipitate, generally as explained in the previous, antibodies are bivalent and antigens are naturally occurring antigens are polyvalent, they are multivalent. So, antigens, they must have polyvalent, here refers to the different epitopes. If they are different epitopes, they react with the different antibodies that are present in the serum. The types of precipitation reactions are the precipitation reactions can be carried out in solutions and in agar gels and the gels with an electric field. If the precipitation reactions are carried out in solution, the examples for such tests are ring test and flocculation test. If the precipitation reactions in the gels, that includes 4 types, that is a single diffusion and in one dimension. This is based on the number of reactants that are diffusing and the direction of the diffusion. Because single diffusion and double diffusion, 1 dimension and 2 dimensions. If the precipitation reactions in the gels and under the electric field, then these are the types of reactions that would be carried out, that they are immunoelectrophoresis, countercurrent immunoelectrophoresis, rocket electrophoresis and 2-dimensional immunoelectrophoresis. So, first one is a ring test. This is an example for the precipitation reaction that could be carried out in solutions. Test is the simplest form of a precipitation test. In this ring test, the layer of antigen solution is placed over a column of antiserum in a tube. In a tube, the antigen is layered over the antiserum and the precipitate forms at the junction of 2 liquids. The precipitation will be formed at the junction of 2 liquids. Examples of the ring test include the grouping of streptococci by Lansifield. In the ring test, in this test tube, the antigen solution is layered over a column of antiserum and the precipitate will be formed at the junction of 2 liquids. Another one is the flocculation test. The flocculation test can be performed in a tube or in a slide. If it is performed on a slide, then that it becomes a slide test, slide flocculation test. The example for this slide flocculation test is a VDRL test that is used for the identification of organism causing the syphilis. Here in the VDRL test, a drop of each antigen and the antiserum are placed on a slide and they are mixed by rotating the slide. After some time, the floccules will appear on the slide. If the floccules appear, the test is positive. The flocculation test can also be carried out in the tubes. In the tubes, the example for this test is a Corn test. Even this is also used for the syphilis, identification of the organism that causes a syphilis. This is called as a tube flocculation test. Tube flocculation test can also be used for the standardization of toxins and toxoids. In this method, the serial dilutions of the toxin or the toxoid are prepared and they are added to the tubes which contains the fixed quantity of antitoxin. The tube contains a fixed quantity of antitoxin to which the serial dilutions of toxin and toxoid are added. the amount of toxin or toxoid that flocculates optimally with 1 unit of the antitoxin is defined as a lethal factor dose. So, like this the toxins are standardized by using the tube test. The example for the tube tests include the Kohn test and the standardization of toxins or toxoids. Next one is precipitation in agar. The precipitation in agar includes the single diffusion in one dimension. Precipitation in agar is also called as immunodiffusion. The precipitation reactions, if they are carried out in an agar gels, they are called as the diffusion, because in the gels the antigens and antibody they both diffuse towards each other. So, that is why this is called as an immunodiffusion technique. Here the agar is generally the concentration of 0.3 to 1.5% is used. Agarose is preferred than the agar because there won't be, agarose do not have any charge, whereas agar has a negative charge. So, there won't be any reaction with the gel, there won't be any reaction of the components with the gel. So, agarose is preferred than the agar, and these are again classified based on the number of reactants that are diffusing and the direction, that in which direction that they are diffusing. Based on the direction and number of reactants, then they are classified into 4 types. They are single diffusion in one dimension, and this is developed by Ordin, and another one is a single diffusion in 2 dimension. This is also called as a radial immunodiffusion, and the other one is a double diffusion in 1 dimension, and the other one is a double diffusion in 2 dimension. These are the 4 types based on the diffusion of the antigen and antibody and the direction of diffusion. And the advantage of these gels over the liquids is because the reaction here it is clearly visible as a band of precipitation. The advantage of gels over the solutions is reaction is visible as a band or a line of a precipitation, and this is stable and can be stained for preservation if necessary. And each antigen antibody reaction gives rise to a line of precipitation. Precipitation line will be produced at the reaction of antigen and antibody. So, the number of different antigens in the reacting mixture can be observed. Immunodiffusion can also helpful to identify the identity and the cross reaction and the non-identity between the different antigen. Types of precipitations in agars are 4 types. The first one is a single diffusion in 1 dimension. In this method that was pioneered by Ordin, he was the one first time used the gels for precipitation reactions. To the antibodies are incorporated into a agar gel. This is an agar gel, and in the agar gel the antibody is incorporated in a test tube. Agar gel with the antibodies. The antigen solution is poured. Antigen solution is poured. The antigen diffuses downwards through the agar gel and forms a line of precipitation. The number of bands indicate the number of different antigens that are present. So, in this single diffusion in 1 dimension, the antibody is incorporated in the agar gel, and that agar gel is placed in the test tube. Antigen solution is layered over the agar gel. Now, the antigen diffuses downwards through the agar gel and it forms a line of precipitation when it reacts with an antibody. This is the number of bands that are produced that indicate the number of different antigens that are present. Other one is a single diffusion in 2 dimensions. This is called as a radial immunodiffusion. Radial immunodiffusion, this is a single diffusion in 2 dimension. Antiserum is incorporated in the agar gel, and that agar gel is poured on a flat surface, whether it may be a slide or it may be a petri plate. So, the agar gel that is containing the antibody is poured on a wells are made. These are the wells. Now the antigen is applied into the wells. Antigen is added into the wells. Now the antigen, it diffuses it radially in all the directions from the well, and it form a ring shaped bands of the precipitation around the well. The diameter of the halo, that is a halo, the diameter of the halo gives rise to the concentration of the antigen. The greater the amount of the antigen, then the farther the ring from the well. So the diameter of the ring is directly proportional to the concentration of antigen. So this method is used for the estimation of immunoglobulin classes in serum and the screening the antibodies to influenza viruses. Radial immunodiffusion, here the antigen diffuses radially the well and forms a ring shaped bands. The antibody is already incorporated in the agar gel, and that agar gel is now poured on a flat surface. Now the wells are cut, and to the wells the antigen is added, and the antigen now diffuses from the wells and it form a ring shaped bands. If the diameter of the ring is directly proportional to the concentration of the antigen, if the diameter of the ring is small, then it indicates the concentration of the antigen is less. If the greater the amount of an antigen, the ring will be the farther from the well. In this picture, there is a increasing an antigen concentration. So in the first one, the diameter of the halo is very less, and in compared to the diameter at the fourth one. So this is used for the estimation of immunoglobulin classes in the serum, and also for the screening for the antibodies to the influenza viruses. Other one is a double diffusion in one dimension. This double diffusion in one dimension, here it was developed by Oakley-Fulthorpe procedure. This is also called Oakley-Fulthorpe procedure because it was developed by Oakley-Fulthorpe. In this, the antibody is incorporated in the gel in a test tube. Here this test is again carried out in a test tube, and here the agar gel is placed in which the antibody is already incorporated, and above which the layer of plain agar is placed, and after the plain agar the antigen is laid on the top of the plain agar. In this procedure of double diffusion in one dimension, here in the test tube the agar gel containing the antibody is placed, above which a layer of plain agar is added, and the antigen is layered on the top of the plain agar. Now the antigen and antibody, they move towards each other through this column of the plain agar. If it is an agar gel plus antibody, and here it is in plain agar, and above which is an antigen. So now the antibody and the antigen, they move through this intervening column of plain agar and form a band of precipitate where they meet at the optimum proportions. Antigen and antibody, they diffuse, that is why this is called as a double diffusion. Another one is Ouchterlony Immunodiffusion. This is a double diffusion in two dimensions. Generally, this Ouchterlony Immunodiffusion is used to compare the different antigens and the antisera. This is the widely used method for the comparing the different antigens. And here the agar gel is poured on a slide. If it is a slide, the agar gel is poured on a slide and the wells are cut. The antiserum, here the antiserum is placed in the central well. This is a central well in which the antibodies, means the antiserum is placed in the central wells, and the different antigens are placed in the surrounding wells. If the two antigens are identical, then the lines of precipitate will be formed by them will fuse. In the first case, it is a non-identity. It is a central well containing the antiserum with antibodies, and the antigens are placed. Here the antigens are A and B. So they are non-identical. So when the non-identical antigens are placed, the both antigens, they will cross to each other. They will cross each other. That indicates that these two antigens are unrelated and they are non-identical. But here if you see the antibody, it is an A, and the antigens here, they are A and A. Both antigens, they are similar. So when precipitin lines, they fuse to each other, the precipitate formed by them will fuse, and that indicates that these two antigens are identical and they are related to each other. Partial identity, the antibodies, they are the antibodies in the central well, and the antigen is A and another one is AB. So A and A, that means partially they are identical. That leads to a formation of spur. In this method, both antigen and antibody diffuse independently through the agar in two dimensions, horizontally and vertically. This test is performed by incorporating the antibodies into the agar, and that is placed on the slide or the petri dish. The agar gel is first poured on the glass slide and the wells are cut, central well and the surrounding wells. The antiserum containing the antibodies is placed in the central well and the different antigens are added to the wells surrounding to the central well. That is incubated for about 12 to 48 hours in a moist chamber, and the line of precipitants will be formed at the sites of the combination of an antigen and antibody. So generally 3 reactions will be observed. If they are non-identical, that means they cross each other. The precipitin lines are fused and it forms an arc, then it is called as an, that indicates that the two antigens are identical. If it leads to a formation of a spur, that means one cross the other, that shows the partial identity of the epitopes. Next one is immunoelectrophoresis. Here the precipitation reactions are carried out in the agar under the electric field. The precipitation reactions, as explained, they can be carried out in the solutions. The examples are ring and flocculation test, and the precipitation reactions in the agar, they are single diffusion, double diffusion, and the third one is immunoelectrophoresis. Here the precipitation reactions are carried out in the electric field. In this immunoelectrophoresis, it is a process of combination of immunodiffusion and then electrophoresis. Immunodiffusion and the electrophoresis are combined in this process, that is why this is called as an immunoelectrophoresis. It is a method in which first different antigens in the serum are separated under the electric field based on the charge. In this method, first agar was poured on the glass slide and a well was made. In the well, the antigen was placed and the electric current is passed through the agar. Now the well is filled with the antigen. Now under the electrophoresis, based on the charge and the size, the antigenic proteins, they move. Following the antigenic separation under the electric field, a trough is cut. This is a single line, this is a trough. A trough is cut and into the agar and it is filled with the antibody. Now the diffusion is allowed to occur as the antibody and the antigen, they diffuse toward each other and they form a series of lines of precipitin. These lines, they are the lines of precipitation. The advantage of immunoelectrophoresis is that number of antigens can be identified in a serum, and the total number of antigens that are present in a serum can be identified, and this method is used to detect the normal and as well as the abnormal proteins present in the human serum, such as myeloma proteins which will be present in the abnormal conditions in the humans. Immunoelectrophoresis, it's a combination of immunodiffusion and electrophoresis. In this method, an agarose gel is poured on the slide and the well was cut and the well is filled with an antigen. Now the antigen is subjected to an electric field. The antigens are separated by means of an size and their charge and the electric field. Now the trough was cut to fill the antiserum. So that is kept for incubation. The antibody and antigen, they move towards each other and they form a precipitin lines. The advantage of this is the number of antigens present in the serum can be identified using immunoelectrophoresis. Other one is countercurrent immunoelectrophoresis. Countercurrent immunoelectrophoresis, it depends upon the movement of antigen towards the anode and antibody towards the cathode. Agarose gel is poured on the plate or the slide. Now the wells are cut and to the wells the antigen and the antibody are added. Now both the antigen and antibody, they diffuse and they move towards each other. Only two wells are cut in this. One well is filled with an antigen and the other with an antibody. Electric current is passed through the gel. The migration of antigen and antibody occurs in the electric field and the line of precipitation will be formed and that is visible within 30 to 60 minutes. So it depends both on the movement of antigen towards anode and antibody towards cathode. Test is performed on a glass slide with an agarose gel. In the agarose gel, two wells are made. One is filled with an antigen, another one is filled with an antibody and the electric current is then passed through the gel. Now the both antigen and antibody, they migrate towards each other and form a precipitation line that is visible in 30 to 60 minutes. This countercurrent immunoelectrophoresis is 10 times more sensitive than the standard double diffusion techniques. This has an applications in detecting the various antigens and it has clinical applications in detecting the various antigens such as alpha feto protein in the serum and the antigens, specific antigens of cryptococcus and meningococcus in cerebrospinal fluid. Another one is rocket electrophoresis. Main application of this rocket electrophoresis is quantitative estimation of antigens. Quantitative estimation of antigens, and it is an adaptation by radial diffusion, radial immuodiffusion, which was explained earlier. And here the rocket name is derived because of the shape of the precipitin bonds that would be appeared in this process. The precipitin bands will be in the shape of a cone, so that is called as a rocket shape, that takes the shape of rocket. That's why this electrophoresis is called as a rocket electrophoresis. Here in this, the antigen in increasing concentrations is placed in the wells in the agarose gel. In the agarose gel, wells were made, and in the wells the antigen with the increasing concentration of an antigen is placed in the wells. The antiserum is already incorporated in the agarose gel. In this procedure, the antiserum is incorporated in the agarose gel and it is poured on the glass slide. Now the wells are made and these wells are filled with the antigen in the increasing concentrations. Now this is electrophoresed. The electric current is passed through the gel that facilitates the migration of antigen into the agar. This results the formation of a precipitin line that is in conical shape resembling a rocket. The height of the rocket from the well to the apex is directly proportional to the amount of antigen in the sample. So here the increasing concentration is from here, the cone of the amount of the antigen is less, and here it is, there is an increasing concentration from here to here. So the height of the rocket is directly proportional to the amount of the antigen that is present in the sample. So this rocket electrophoresis is mainly used for the quantitative estimation of antigens in the serum. Next one is a two-dimensional electrophoresis. This is a variant of rocket electrophoresis which was explained, and It is a double diffusion technique used for the qualitative and as well as the quantitative analysis of serum, wide range of antigens. This test has a two-stage procedure. In this test, first a small trough is cut in the agar gel on a glass plate that is filled with the antigen solution. So, in the test, first a small trough, this is a small trough that is cut in the agar gel on a glass plate and that is filled with an antigen solution. Electric current is then passed that lead to the separation of antigens. Antigens now migrate into the gel and depends upon their electric charge and they will get separated. In the second stage, this is in second electrophoresis, the gel piece containing the separated antigens, here this is the gel piece containing the separated antigens is placed on the second glass plate. This is the second glass plate that is placed and the agar containing the antibody is poured around the gel piece. Now the second electric potential is applied. It is perpendicular to the first direction of migration. The direction is from this way. Now the direction of electric force is in this way, just it is a perpendicular. A second electric field is applied at right angles to the first direction of the migration. The pre-separated antigens then migrate into the gel containing the antibodies and precipitate with the antibodies in the gel and lines that will be formed. This method is both qualitative and quantitative. It identifies different antigens that are present in the serum solution. Once again, it is a variant of rocket electrophoresis. It was developed by Laurel and it is a two-stage procedure. 2 times that electrophoresis would be carried out and it is useful for the quantitative and qualitative identification of different antigens that are present in the solution. In this test, first agar gel is poured on a glass slide and a small trough is cut and the small trough is filled with an antigen solution. Now the electric field is applied. Antigens that would migrate into the gel at a rate proportional to the electric charge. The gel piece containing the separated antigen is now placed in second glass plate. Agar containing the antibody is poured around this gel piece. Again the electric potential is applied and it is perpendicular and it is right angles to the first direction of migration. Now the pre-separated antigens that migrate into the gel already contained the antibodies and it forms a reaction, leads to a formation of precipitate. Precipitin lines will be formed. When these antigens they combine with the antibodies which are present in the gel. The other techniques include turbidimetry and nephelometry. Turbidimetry and nephelometry are the two methods that are used to detect and quantitate the precipitation reactions in the serum based on the phenomena of light scattering by precipitates. Turbidimetry, as we do in the calorimetric reactions, the turbidity is measured, and the nephelometry, here it measures the light that is scattered when the incident light is passed through a suspension containing the antigen antibody precipitate. So, this is about the precipitation reactions. Briefing about this precipitation, the soluble antigen reacts with the antibody in the presence of electrolytes at suitable temperature and pH that leads to a formation of insoluble precipitate. If the precipitates are not sedimented and they are suspended, then it is called as a flocculation. These precipitation reactions, the mechanism was explained by Merak. Proposed the lattice hypothesis at the zone of equivalence where the antigen and antibody concentrations are optimal, that leads to the formation of lattice, and in the pre-zone and pro-zone there won't be a precipitate formation because of the excess amount antibodies and the antigens. These precipitation reactions can be carried out in the solutions. Examples is ring test and the flocculation test. Flocculation, it can be carried out either in the slides and the tubes. Now the precipitation reactions can be carried out in the gels. Agarose is preferred than the agar. So agarose gels that is in the single diffusion in one dimension, that is odd in single diffusion in two dimension. This is radial. Here in the radial diffusion, body is incorporated in the agar, and that agar is poured on the slide, and the wells are cut, and the wells the antigen is placed. Now the antigen diffuses and it forms a ring around the well. The diameter of the halo, it is directly proportional to the concentration of the antigen. And the next one is a single diffusion in two dimensions, and the other one is a double diffusion in one dimension. Double diffusion in two dimension, that is Ouchterlony diffusion. Here this is generally widely used for the comparison of different antigens and antisera, and both here antigens and antibodies, they diffuse independently, and if antigens are identical, that leads to the formation of a arc, and if they are non-identical, they cross over each other, and if they are partially identical, it leads to a formation of a spur. Precipitation reactions can be carried out in the electric field in the gels, then that is called as a immunoelectrophoresis. And in this immunoelectrophoresis, first the antigen is separated under the electrophoresis, and the trough was made. To the trough, antibodies are added, and the antibodies and antigens now they diffuse and form a precipitation lines. In the countercurrent immunoelectrophoresis, on the glass slide, agarose is poured on the glass slide, and two wells are made. One is filled with an antigen, another one is filled with an antibody, and the electric current is passed through them, and these move towards each other and form a precipitin line at the junction. That is a countercurrent immunoelectrophoresis. Another one is a rocket electrophoresis. It is an adaptation by radial diffusion. Here, first antiserum is incorporated in the agarose gel, and it is placed on the glass slide. Now the wells are made, and the wells to the wells the increasing concentrations of antigen is added, and that is both antiserum and antigen is subjected to electrophoresis. It leads to the formation of a cone shaped structures, and the height of the cone is directly proportional to the concentration of the antigen. In the two dimensional, it is a two stage procedure. Both it can be used for qualitative and quantitative. In the first electrophoresis, the antigens are separated. The separated antigenic piece is now placed on a second plate. The second stage, the gel piece containing the separated antigen is placed in the second glass plate, and the agar, that is agar containing the antibody, is poured around this gel piece and subjected to second electrophoresis, that is perpendicular. It is right angles to the first electrophoresis. Now the pre-separated antigens, then they migrate into the gel and form a precipitation with the antibodies. This will be the precipitation lines. This is about the precipitation reactions. In video, I will explain the agglutination reaction, other antigen antibody reactions. Thank you for watching this video.